everyone, and welcome to Her Science. Today we are going to be talking about feedback inhibition. We are going to be going over the definition of feedback inhibition, as well as some helpful terms. We're going to be going over why is feedback inhibition even important, and we're going to be going over this diagram that I drew out for you guys. But first and foremost, what is feedback inhibition? Feedback inhibition is a mode of metabolic control in which a metabolic pathway is halted by the inhibitory binding of its end product to an enzyme that acts early in the pathway. Now, this is a big, scary definition for a really simple process, which you guys will see in the diagram I drew out. But first, some helpful terms. You guys definitely need to understand what these are in order to understand what feedback inhibition is. Metabolic pathway is the sequence of enzyme-catalyzed reactions that lead to the conversion of a substance into a final product. Pretty much, it's the path or the road a substance takes to become its product. And it's that there's typically several chemical reactions that occur that are catalyzed by enzymes during that pathway. Now, an enzyme is a molecule that catalyzes or speeds up a chemical reaction. Something that's really important to note here is that enzymes are not consumed in a reaction. They just help out the reaction. And something else that I'm going to write here as well, they are shape specific. Okay. And I'll explain that further in our little diagram here. And then here we have why is feedback inhibition even important? I'm going to talk about this after our example. So we're going to start here at step 1. So we have our generic substrate. This is the beginning of our metabolic pathway. In step 1, our substrate bonds with our first enzyme here. When they bond, they create this enzyme substrate complex. And in this complex, a series of chemical reactions occur. And the enzyme spits out this molecule here. I've named it intermediate. Now, like we just said, enzymes are not consumed in a reaction. So our enzyme 1 is just going to hang out up here. Now we are going to introduce enzyme 2. To help turn our intermediate into our product down here. So our intermediate and our enzyme 2 bond, and they form this enzyme substrate complex here. And again, a series of chemical reactions occur, and it spits out our product. So now that we have our product, this product is distributed throughout the system, or say our bodies. Say our bodies need a whole bunch of this product for whatever reason. The product is distributed throughout the system as needed until no more is needed. That's important. It's distributed until no more is needed. Now we're on step 7. When no more of the product is needed, the last product made bonds with enzyme 1. So here we have enzyme 1. Our product bonds with it. And when this happens here in step 8, this bond changes the shape of enzyme 1, which stops it from being able to bond with new substrate, therefore stopping the synthesis of the product. So our product, there's no more that's needed in our body. So this product is just kind of hanging out with nowhere to go. So it bonds with enzyme 1, and when this bond occurs, the shape changes. So remember how we were saying that enzymes are shape specific? Because the shape changed, this is enzyme 1 as well, our original substrate can't fit into here anymore because it's now changed to this shape. So say some time has passed and our body, for whatever reason, needs more of this product. Our product here is going to leave our original enzyme, and it's going to be distributed to the system, or our bodies in this case. So when the system needs more product, it leaves the enzyme, allowing the enzyme to return to its natural shape. The enzyme can now return to step 1 and begin accepting new substrate. So we see here, it's back in its original shape and can begin the whole process over and over and over again until our body no longer needs this product any longer. And then we return, to step 7, where it bonds with enzyme 1. So why does our body do this? Why is feedback inhibition even important? So it prevents cells from wasting energy and substrates on chemical reactions that are not necessary at the time. So I'm sure you guys have been introduced to energy and ATP at this point. Our bodies only have a limited number of ATP molecules at any given time. So if a bunch of ATP molecules are being used for chemical reactions that don't even need to be happening, then there's not going to be enough ATP molecules to provide energy for chemical reactions that are necessary for life at that moment. And if you've learned anything about evolution, then you know that evolution favors efficiency. So here arises feedback inhibition. That is why it happens. Thank you guys so much for watching. If you have any questions, leave them down in the comments and I'll try to get back to you guys. But other than that, thank you guys so much for watching and I'll see you next time.